Since there is no more doubts.
Hello, can I have a question?
Yes,
Yes, you
please,
are
madam.
audible.
Hello.
Yes, you are audible.
Yes.
Yeah. See, I'm professor from Tamil Nadu Teachers Education University, Professor Kanmani. And this is, sir, regarding your presentation. You have spoke about the status of research and your findings, whatever you have done in foreign countries. But the countries like India, and especially the cities like the cosmopolitan cities, where you could find a lot of pollutions and everything. I think you might have heard about the pollution, the status of pollution, especially air pollution in Delhi. So, if that is the case, and what is your opinion or your idea to produce some of the vegetables like cauliflower and broccoli, and this is what I can see, and grapes, and whether it is seed or seedless, where we can find a lot of pesticides have been used. So, but still, we know the limitations or the side effects of using pesticides on those fruits and vegetables. And what can you suggest as a green pesticides, or is there any findings which can be used alternative pesticide instead of chemicals?
Okay. First of all, thank you, madam. Thank you very much. And it's a very nice, wonderful question. And particularly, actually, this is why now the existing organic, in our place in India, we always people are now very, very much fond of organic farming and everything. Even the food item and everything, people are saying organic. So organic means here we are not going to use any chemical insecticide, but already, as in the presentation, I have already mentioned that we are the people, we always want the immediate result. So that's why we are always preferable chemical insecticide, because you will get the result immediately. But in the recent days, but I have found in India, even from Tamil Nadu and other parts of the world, other part of the India, people are using a lot of alternative source like cow urine and what's it called, panchakavyam or something. They are preparing as an insecticide. Even even nowadays, even people are using neem oil as insecticide in order to act as a anti-feeding activity, because it will drive away the insects from the plants. Drive away the insects from the plants. So this way you can use some kind of, but the effectiveness always again it is a questionable when compared with the chemical insecticide. The plant derivatives one are, they may be effective, but you will not get the results immediately. But you have to do it continuously, more than 2 years, 1 year, then only we can develop the one. This is where the scientific, the reason, the problem is existing. We have to convert our natural resource as a very potential chemical like a chemical insecticide in the near future. So there is a bioinformatic tools and biocomputing technologies, and everything is around the corner right now. So we are on the way, and probably in the near future, we will get the most potential insecticide, just like a pyrethroid, even from the some other plants. I hope in the near future we'll get it, madam. Thank you so much.
Sir, is there any statistical data we have based on any research findings that how much of organic farms are being used when compared with the chemical, that is the modern chemical pesticides?
Okay, but it's very meager amount. It's maybe less than 2% or something. Even you can see, I am from the village, from Tamil Nadu. So where I can see only one if there are 1,000 farmers are there, only 1% he will go for the organic. So it may be less than 1%, or it may be less than 2%. But the reason is we cannot blame the farmers, because the farmers he want to enhance the crop yield. So he need immediately the fertilizer and the chemical insecticide in order to in order to control his pest. But we are suggesting them to go for the plant based, but the plant based are ineffective when compared with the chemical insecticide. So
Okay,
this
but
way
what can
we.
you? Yeah, okay. But the thing is, I think I hope you're in abroad, but in the sense, how these things have been used, practiced in the organic farming, or practiced in abroad. Uh, those technology can be transferred to India, or can you suggest any kind of technologies which can be used to increase or enhance the organic farmings by the government, or could you suggest any steps to the government?
Yes, yes, I can suggest. Actually, I have visited many European countries and even the Kingdom of Saudi Arabia and other parts. What they are doing, greenhousing. They're doing greenhouse mechanism, and it's not possible in India because our climates are very good to cultivate, to plant crops and everything. Suppose you take example, Saudi Arabia, 45 degrees centigrade. So here you cannot grow the plants. So what they are doing, greenhouse they are making. So with the green sheet and everything, so they are cultivating the crops and even the vegetables and everything under the roof. So that is a closed one. So it is a very closed one. So there will be the the insects cannot be damaged very easily there. So they are adopting here the conventional, that is called the plant-based insecticides and other things in order to control insects. But if you take it Kerala, or if you take it Tamil Nadu or the rest of the India, we don't have very limited, only for the experimental purpose maybe we do greenhousing farming. But otherwise our farmings are very much open. If you take my village, more than 5,000 acres or 10,000 acres are full of rice field. So suppose what I will do, suppose I have a 2 acre of land and I am going for the organic farming. But my neighbor farmer, my neighbor, what he is doing, he is a chemical insect. He applying the chemical insect. So what will happen? Automatically, when he apply the chemical insecticide, automatically he drive away the insects to my field because I am using organic form and I am using only plant-based pesticide and it may not be effective. So automatically the organic farming will be spoiled here in India. This is the correct
That's
scenario.
what my question is actually. What is your
Hmm.
suggestion to the government as a scientist,
Hmm.
so that these kinds of chemical pesticides can be avoided?
Yes,
At
actually,
least
based on
slowly.
this,
At least slowly.
yes, yes. Actually, for the government, this is what I am suggesting you. When compared with the earlier, now lot of neem based. That means vapamaram. Neem based, lot of insecticides are derived now, azadirachtin or something like that. So there are available. But when compared with the chemical insecticide, but it is very it is very less percentage, maybe less than 10% we are using for in the farming activities, and this is why, and we have to go for the this one. Actually we have to develop a novel innovative insect control technology by using plant as a pesticide, by using available technologies, available computational and information technologies. We have to develop a very potent insecticide in the near future. For that, we need all the pertinent stakeholders must be brought in the same platform by the policymakers. And apart from this one, our curriculums also has to be designed in such a way that, because as I mentioned already, we do lot of research. We spend a lot of more than 4 years, 5 years for PhD program, MPhil program 1 year, and MSc dissertation and everything. And finally, it end up with a one dissertation work or one PhD thesis, one research, 2 or 3 research paper. That's all, and never we convert them into the practice. It's not only in the biological science. I am saying in all the field. This is what I have observed. Maybe one few people they will get the pattern and everything, and they are using as a in the common man usage. But otherwise, more than 98% of our research are simply is lying in the library as a research work, as a research paper, or thesis, or a dissertation. That's all. So the policymaker has to make a decision. The policy must be designed, and all the curriculum must be designed in such a way that in order to enhance the plant-based insecticides as a potential like a chemical insecticide in the near future.
Okay, sir. Thank you.
Thank you so much.
Just one question, one more question can be asked. Just one more, a brief, if. Okay, I assume that there are no more questions, so it's time to wind up. See, the scientists are trying to enhance the health and wellness of the people globally. But at the same time, there are many things happening around the world which affect the health and wellness of the people. Right? War is going on everywhere. Diseases are spreading. Pandemic kind of diseases spreading. So I was shocked to read today's newspaper that the newly elected President of United States of America, Donald Trump, decided to quit from WHO. So the effect could be anything. We don't know at this stage, but it's quite alarming for the reason that it is the United States that funds 75% of the WHO activities. So if they quit, so who is going to fund? They are going to suffer it. Let's hope so. So global politics actually affect human health and the happy living of humans worldwide. So today's talk was really inspiring. Dr. Karunamurthy talked about the vector-borne diseases and its control. Basically, I'm a parasitologist. I work on helminth parasites. So I'm well aware of how important the vectors are and how important it is to control the vectors, because helminth parasites also have vectors, the snail vectors. And you also explained the malarial parasites. Of course, I don't work on protozoan parasites. Still, it is the mosquito that serve as a vector. So he discussed vector-borne diseases and their control. And mosquitoes are one of the leading vectors of human diseases. And he also discussed the tropical climate that favors the spreading of the rapid multiplication of mosquitoes. And I have personally observed the rapid swarming of mosquitoes, particularly in North Indian conditions. And he also explained the limitations or problems of using chemical pesticides. And we cannot continuously using it. We need to find some alternative, some plant-derived or natural products as an alternative to these chemical pesticides. So hopefully, as some of the audience asked the question that are we able to completely stop by using these kind of products? The question is no, but we are on the right track. Hopefully, we will be able to find some naturally available or plant-derived products that can control these vectors. If not fully, at least partly. So in that way, we can at least reduce the use of these chemical pesticides and save the health of humans globally and regionally. So it was a wonderful talk on behalf of the organizing committee. Let me thank you for finding time out of your very busy schedule. So let me invite Dr. Aisha Banu to deliver the official vote of thanks. Thank you, sir.
Most welcome, sir.
Good afternoon, everyone. I would like to take a moment to express my heartfelt gratitude to each and everyone who being part of the session. First and foremost, I express my sincere thanks to our keynote speaker, Dr. A.K. Karunamurthy sir, for the time and effort you have invested in sharing your knowledge with us. Thank you, sir. Thank you very much, sir, for your wonderful session. Next, I would like to thank our session chair, Dr. Ahmed Sharif sir, for chairing the session. Thank you, sir, for your inspiring comments. Finally, to all online and offline participants, research scholars, delegates, colleagues, and students assembled here, I express my sincere appreciation for your active participation, questions, and discussion, especially Kanmaniman. Once again, thank you very much for making the session a great success. Thank you all.
Okay, madam. Thank you very much for giving the time and this opportunity. And good luck for the rest of your conference and everything. And thank you so much. God bless you.
Thank you, sir. It's
Thank you,
time
sir.
to
Thank
wind
you.
up this session.
Okay,
Thank you
thank you, sir.
all. Thank you, all the participants.
Nowadays it is difficult to change our climate conditions. So we have conducted this experiment for low irrigation areas, how to cultivate these plants that kind of areas. What will be the future prospect of the business? It will extend this work to the areas of this water deficit. Low irrigation or water stress conditions, it will extend this work. You can do the work in a similar way. You have done the germination analysis only. So it is a tropical plant. Usually, we used to plant and water during germination. The growth parameters and the yield could also be noted. So this study could be done in the long run, for all parameters, in order to analyze all the drought, stress and stress, effect of the stress on this bird. Nice to know all this. Thank you, Marwa. Move on to next session. I call Chaitra K. Chaitra,
Yes, ma'am.
are you audible?
I'm present now, ma'am. Hello. Hello. Can I start?
You can start.
Hello.
You can start now.
Okay, ma'am. Okay, ma'am. I'm Chaitra. I'm a PhD scholar from Sarset College. Today, I'll be discussing on the topic Unlocking the Potential of Wild Edible Plants, a Sustainable Approach to Nutrition and Health. This study is about the wild edibles' potential for food security and biodiversity conservation. So, what are wild edible plants? Wild edible plants are plants that grow naturally without the intervention of the human action. So, in the right side, you can see the figure which depicts the history of wild edible plants. That is, in the ancient times, how this ancient people depended on the wild edibles for their daily needs. As agricultural revolution happened, their dependency have been shifted towards this monoculture. As again and in the right side, you can see the modern revival. That is again there comes an interest towards this wild edibles. So this is a history of wild edible plants. Why wild edible plants matter today? What is its significance? As it is nutritionally richer, you can see in the coming slides. So it is culturally important. That is, it is used by the indigenous people for various purposes like food, medicine, their religious practices, everything they are using. So it is culturally important. It is environmentally, it plays an environmental role. That is, it supports biodiversity. How it supports biodiversity? Because is it as it growing naturally, it does not require any fertilizers, pesticide, or anything. So it gradually the pollution will be pollution will not be there, and as people are more aware of this wild edible, they will be conserving this wild edible. So again, this biodiversity will be conserved. So it reduces the carbon footprints, climate mitigation. That is, climate change mitigation can be done. So these are the environmental role. Research overview. I have done this research among the Pania tribes, which is the largest scheduled tribe of the Kerala district. So it is so they are mainly found in the Wayanad district. So I have documented the wild leafy greens used by this Pania group, and from them I have shortlisted 3 plants and done the nutritional analysis and compared it with the cultivated greens. So research methods include field survey with the Pania people led into the documentation of 64 wild leafy greens, and from that Hibiscus hispidismus, Laposia interrupta, and Marsilia cordifolia have been taken for the analysis in this study. So it is being checked for protein, carbohydrate, fat, and fiber using this respective methodology. Nutritional composition of wild edible plants. You can see in the left side of the slide that is showing the nutritional composition of the respective plants. So in the right side, you can see that Hibiscus hispidismus is having the highest quantity of protein as well as fat. It is also having the highest quantity of carbohydrate, but similar to that of Marsilia. Marsilia is having the highest quantity of fiber. So nutritional composition of commercially cultivated grains. You can see in the left side the table showing the nutritional composition of the respective cultivated greens. So from this graph, you can find out that amaranthus is having, which is shown in the blue color, is having the highest quantity of protein as well as fiber. But in case of carbohydrate, cabbage is having the highest quantity of carbohydrate. And in case of fat, spinach is having the highest quantity, but similar to that of amaranthus itself. So what I have done is that I have compared these two groups, and from the graph itself we can identify that these wild edibles are richer in case of these nutrients. Every parameters they are richer. So this marks the importance of wild edibles. Proteins are building blocks of the body. Carbohydrate, it sustains energy levels, that is primary source of energy. Fats, it is used for fat absorption of fat soluble vitamins and energy storage. Fiber, it supports digestion and prevents constipation. So of course, as these plants are nutritionally richer in these cases, so it have a nutritional significance. So socio-cultural and environmental impacts. As we know that it is these plants are used by these groups, these indigenous groups. So it is culturally important, environmentally benefits. That is, it grows naturally, so low maintenance, no need of fertilizers or pesticides. Food security, as it is nutritionally richer, it can tackle this food scarcity. It is used for food security. So sustainability, it supports biodiversity and reduce the reliance on the cultivated crops. Wild edible plants are sustainable solutions. Almost 8 lakh of children under the age of 5 is suffering from malnutrition, that is stunted growth. So you can address this malnutrition using this wild edible plant. Sustainability, as it grows naturally, we know that it does not require anything like fertilizer or pesticide. So carbon footprints are reduction in carbon footprints. So food security, it provides alternates during crop failure. What is the challenges and recommendation? Challenges include loss of traditional knowledge, change in diet, urbanization, landscape change, access and availability reduction. What is the recommendation? Awareness campaigns, integration into food systems and policy making, community training programs, support conservation and sustainable use practices. So these are the recommendations for protecting these wild edible plants. Wild edible plants a path forward. What is the takeaway? As it is nutritionally richer, of course, food security, sustainability, cultural significance, addresses malnutrition and biodiversity conservation. So we have to embrace this wild edible plants for a sustainable future. So this is the call to action. So we have our Pioneer College have started a blog known as Epic Pioneer College, which documents this ethnobotanical plants in the college. So we have also started a nursery for these ethnobotanical plants. So finally, I'll be concluding by saying that a leaf for sustainability, a shield for food security. Thank you so much for hearing me.
A nice presentation, by the way, Chatramas.
Thank you, ma'am.
Okay. Have you gone through the conservation status of these particular plants?
Conservation.
I mean,
Actually,
is it belong to,
what, ma'am?
does it belong to an RET category or something?
No, ma'am, not RET category.
Not RET category. Okay.
No,
So
no.
you have gone through the nutritional aspects only.
Yes,
so
ma'am, nutrition
wild
aspects
plants-
only.
These wild plants might be having other components also, which is, you have done the secondary metabolite analysis.
Yes, ma'am, everything I have done, like secondary flavonoids, phenolics, tannins, everything I have done, ma'am. Saponins, cardiac glycosides, everything. But only here I've shown a little one only, ma'am.
Okay, is there
So
a
it is
presence of
having that much of
toxic components?
like it phenolics is no, ma'am, no, ma'am. Toxic, I have checked for anti-nutritional components like tannins, but it is only lesser in amount, so it is not a problem, ma'am.
What about alkaloids?
Like alkaloids is almost greater in content, so we can go for this secondary metabolites like cancer treatment. We can use that. Like I have an idea. I don't know whether I can do it right now, but after this PhD, I'm going for this. I would like to go for cancer study or like that pharmacological study. I would like to bring these plants, ma'am.
Okay, is the quantification of these particular components done?
Yes, ma'am. Yes, ma'am. Sure, I have done that quantification, ma'am.
Quantification. So if it is having high
yes,
quantity
ma'am.
of alkaloids, it won't be able to use as a nutritional component. That's why. That's why.
Okay, ma'am.
Okay. Can you predict the result of prolonged use of these plants?
Like I have said, like it is nutritionally richer, so obviously we can tackle this malnutrition. Like we can bring this wild edible plants into concern, so we can make we can of course address this food security, ma'am.
Okay. Along with that, if the secondary metabolites, including the anti-nutritional components, if on prolonged use, it might get accumulated inside the body, right?
Yes, ma'am, but it is not in higher quantity actually. So it will not be a problem. Like in case of cultivated crops also that is having a little bit of like that, like we can compare this secondary metabolites with this cultivated crops also. So it is not so higher than that of cultivated actually. But in case of this proximate composition, it is having highest quantity.
So you're recommending the extensive use of these wild plants, right?
Yes, ma'am. Yes, ma'am. Surely, because these people, these tribal peoples are using this, so we have to bring this into our concern also. As we are only knowing about a little plants like amaranthus or chira, cabbage, something we use for daily purposes. But I have documented around one or three plants that is wide leafy edibles itself. So from that, I could understand that after getting into this only, I can understand that this is having, like, we have a wide range of wild leafy greens which we can bring into concern, which we should bring into our implementation, ma'am.
Okay, fine. Just asking out of curiosity, isn't there a chance for over exploitation of these plants and causing instability in the ecological system?
Actually,
So it need to be
eh,
along with promotion. It has to be, propagated also.
yes,
So
ma'am. But we should, like, I have also a concern that ma'am, ma'am, you also have a concern. Like that, I also have concern, but because which it should come into society, but it should be properly managed. Like we should grow for agroforestry, like in case of farming, or we should properly manage it. We should use that. We should. It should be sustainable. That is, this tribal people, what do is that they does not go for younger plants. They just use the mature plants only. Some portion that mature portion only they take. So younger one they keep it for future purposes also. Like that, we should do sustainably. We should harvest them. So it can this proper managing system should be there, ma'am.
okay, are these plants endemic?
Not endemic, ma'am. This some are endemic, ma'am. Some are in this case, this study, this is not endemic, but some the plants that I have documented are endemic to Wayanad, ma'am.
Okay, fine. Nicely defended. Nice presentation. All the best.
Thank you so much, ma'am. Thank you so much.
Hello.
Yes, sir.
One more question. That
Yes, sir.
whether tribal people cultivated this plant or they got it randomly from the forest?
Actually, no. Actually, this Paniya people, I have done in many tribal groups. That is Paniya, Kuruchia, Kuruma, Adia, every tribal groups. Like almost 5 or 6 groups, I have done this work. But in case of here, I am explaining the Paniya group. So Paniya group is like Paniya group. They usually go for fields. Actually, that is agricultural fields. They cultivate. These plants mostly, that is most of this plant that they use come from this agricultural lands as well as plantations. So some of them they have collected and they have domesticated in their house gardens also. That is kitchen gardens. They are domesticating. Like in case of Sehenaria mysorensis, it is actually alien chappu. We call it as alien chappu. So it is they call it as alien chappu. So it is actually being domesticated. Everything like few example only. One example only I told you, sir. So every plants that they are actually domesticating in their kitchen gardens also, sir.
Okay. Whether you observed that in certain week or certain month, the forest people they used to collect these medicinal plants from the tribal people, especially from your area. Whether you observed that these particular plants are collected for medicinal purpose by the forest department.
A forest department actually, when I was in, like when I was, I had a, when I was going with these people, tribal people, like Kartanaika is the person who is like forest dependent tribal group. So they usually collect. I heard that I have not got that much of idea, sir. But they do this what we'll tell that they collect that honeys or something and bring into markets like that. They have done, but I have not idea of this thing, sir.
Okay. Thank you.
Oh,
Thank you. Nice presentation. Okay.
oh, thank you, sir. Thank you so much, sir.
Thank you, Chaitra. Moving on to next presentation. I call Fatima Najjar for presenting the topic nutritional profiling of Palmyra palm powder and arrowroot powder versus industrial nutritional supplements. Fatima Najjar.
ഇത് 40 കുട്ടിയുടെ ടൈറ്റിൽ ഇതാണ്.
Move to the next presentation. I call Rajla M.
രജത്തിന്റെ രജ
I call Jalva KV for presenting the topic in silico identification of anti-Parkinson's compounds from Centella Asiatica.
ടൈറ്റിൽ എവിടെയോ കുട്ടീൻ്റെ പേര് ജൽവ അല്ലേ? ജൽവ കേവി ടൈറ്റിൽ ഇൻ സിലിക്കോ ആപ്ലിക്കേഷൻ സെൻട്രൽ ഏഷ്യാറ്റിക്ക.
Good evening, distinguished guests, beloved teachers and friends, other fellow researchers who are gathered here. It's an honor to be present here. Myself, Silva, and first year MSc Integrated Botany Application Participant at Mes Keveeyam College, Palangiri. I'm delighted to present my work on the topic identification of anti-Parkinson's compound from the Centella Asiatica through in silico methods. The study driven by the growing global burden of Parkinson's disease, which affects millions of people worldwide, and also it presents a significant therapeutic challenge. So, my aim to contribute to the identifications of potential novel plant-based anti-Parkinson's drugs with aging. There are many symptoms which includes Parkinson's disease, which are motor impairments such as tremor, which can be occurred at rest in the hands and limbs, stiffness of muscle, slow movement, shuffling of gait, and coordination difficulties. There are other non-motor issues like sleep disturbance, cognitive decline, speech problems. All these symptoms collectively impact the mobility, daily functions, and overall quality of the life. So let's look into the global statistics of Parkinson's disease. According to WHO, in 2019, there were over 8.5 million people who live with Parkinson's disease worldwide. And in India, which forms to nearly 0.58 million people living with Parkinson's disease as an estimated in 2016. So these estimates highlight that these statistics highlight the need for the study or need for the innovative approaches to address these challenges. So the needs of the study include which there is no effective cure and the limited pharmacological alternatives are available. The current therapies only provide symptomatic relief. The PD medication can cause serious side effects like other synthetic chemical medicines. Administration of levodopa, which is the only known medication of Parkinson's disease. Many medication like and surgical resources which are not accessible to particularly low and middle income countries. So I will outline my objectives, which are identification of potential target protein associated with Parkinson's disease, evaluate the anti-Parkinson's activities of phytochemicals which are derived from Centella Asiatica through in silico methods, and analyze the protein ligand interaction. Also to analyze various properties of phytochemicals like molecular properties, drug likeness, and pharmacokinetics. To establish these, we established a robust methodology, which is the first step in the methodology is the identification of target protein. The very next step is to identify the active sites using PDB-SUM server, and which is followed by the preparations of phytochemicals which are already reported from the Centella Asiatica. Then the most crucial step, which is the molecular docking process, which is achieved through virtual screening procedure. And the results were first analyzed by means of binding affinity scores. Then further post-docking analysis was performed to study the protein ligand interactions, and other studies like molecular properties, drug likeness, and pharmacokinetic properties are also analyzed. The success of this in silico study depends upon the tools and crucial tools and materials we used. The monoamine oxidase, which is the target protein we use. Monoamine oxidase facilitate the oxidative deamination of neurotransmitters including dopamine, serotonin, epinephrine, and this inhibitor functions by preventing the breakdown of these neurotransmitters in the brain. MAOs are class of flavoproteins. Here is the structure of monoamine oxidase from the PDB site, and its PDB ID also mentioned, which is 6FW0. This structure contains 2 chains, each of 250 amino acid length. The next is the phytochemicals from the Centella asiatica. It's an important medicinal herb which is commonly called gel trummy. It is used in medicines of Ayurvedic traditions in India for thousands of years, and also listed in historic Srusut Samhita, which is an ancient Indian medical text. Centella asiatica belongs to Umbelliferae family. It is found throughout India, growing in the most moist places. The most important, Centella asiatica has a diverse and complex chemical constituent which includes terpene, which may be monoterpene, sesquiterpene, triterpene, or tetraterpenes, and phenolic compounds, polyacetylene groups, alkaloids, carbohydrates, mineral acids, and amino acids. Because of the presence of these diverse complex constituents, these also exhibit various pharmacological properties like anti-inflammatory, anticonvulsant, antioxidant activities, and neuroregenerative properties. So the next is in silico screening. The molecular docking technique, which is executed via virtual screening procedure. The tool we use is Autodoc Vina. To execute Autodoc Vina, we need 2 requirements, which are the target protein, that is monoamine oxidase, and the ligand in PDB format. After the in silico screening, the post-docking analysis were carried out. The binding affinity scores are studied, and the protein ligand interactions are studied using Discovery Studio Visualizer. The molecular properties, drug likeness score, and pharmacokinetic properties are also predicted. Next, moving on to results and discussions, which is the target protein analysis. The target protein is the monoamine oxidase that I mentioned earlier. Its role is to facilitate the oxidative deamination of neurotransmitters. There is an inhibitor which present in this crystal structure of this target protein, and the active site residues are also mentioned here, which is taken from the PDB site. It is the Ramachandran plot of monoamine oxidase. The virtual screening process here, 95 compounds from the Centella asiatica were screened against the monoamine oxides. The docking score analyzed, and 86 compound shows a docking score less than -6. Out of these 5 compounds, top 5 compounds shows a docking score which is less than -10.8. And molecular property ADME analysis and drug likeness pharmacokinetic properties were also carried out for these top 5 compounds. Based on that, best 2 compounds are selected. These are the protein ligand interactions of the top 5 compounds. Here the 3-epimethylenic acid, taraxosterol, corosalic acid, and promolic acid, urosolic acid. Here the both hydrogen bond interactions and hydrophobic residues are also mentioned. Now, the molecular property analysis of these top-ranked molecules, that is taraxosterol, urosolic acid, corosolic acid, and 3-epimethylenic acid and promolic acid. Here we can see the binding affinity or the docking score. From these, we selected taraxosterol and 3-epimethylenic acid as the top 2 compounds. Even if there are compounds which have higher binding affinity, like 3-epimethylenic acid, based on the ADMET analysis. In ADMET analysis, we figured out that the taraxosterol and 3-epimethylenic acid does not execute any kind of cytotoxicity. Also, it has high number of hydrophobic interactions and good drug likeness properties. So, these are the docked images of the best lead molecule, that is the figure 4.A shows docked complex of taraxosterol and MAO, and its 2D interactions are also represented. And the second is the docked image of docked complex, which consists of 3-epimethylenic acid and MAO, and its 2D interactions are also represented here. So, in conclusion, In the present study, 95 phytochemicals were analyzed for its anti-Parkinson activity through the in silico method. The study revealed that there are 86 compounds which shows less than -6 binding affinity from the top complexes were further analyzed, and the result revealed that the 3-epimethylenic acid and taraxosterol, which have an significant inhibitory activity on the monoamine oxidase with good drug likeness and acceptable pharmacokinetic properties. However, the in vitro and in vivo studies are necessary to support these results. The top compound, taraxosterol, it is an anti-inflammatory, antioxidative, and anti-allergic neuroprotective properties it shows. And taraxosterol is an pentacyclic type, and the 3-epimethylenic acid, it also exhibits antioxidant, anti-inflammatory, hypoglycemic, and neuroprotective activities. It is also an pentacyclic type, triterpene. So, we would like to express our sincere gratitude to Principal of Mes Keveeyam College providing us the opportunity to carry out this work. And we extend our heartfelt thanks to the Head of Department of Botany for their constant encouragement and valuable guidance throughout this endeavor. We deeply thankful to the Head of Department of Bio Retail Management for generously providing the necessary lab facilities to support. Thank you all for your attention. These are some of the references. Thank you. Thank you. Thank you.
Yeah, and I've gotten a really nice opportunity to present your idea. How did you come to Centella?
First, I listed out some of the plants which shows the neuroprotective action from the literature reviews which are available online and like texts. And then understanding the protein ligand interactions or the hydrophobic hydrogen binding interactions. Also, the constituent, that is the phytochemical constituents. I understand that Centella has diverse complex molecules which have such kind of properties. So I selected Centella as the study material.
Ask this because Centella is usually for its wound healing.
Yeah, yes, it has wound healing activity. Yes, yes, yes.
Curious about how did you Centella allow to and what's the duration of the study?
Actually, I think 1 month or 2 to 3 months. That's it. Yes, yes. Actually, in silico docking process doesn't need that much time. That is the advantage. But the analysis part needs much time.
So, from your findings, what are your recommendations for using Centella in
actually, from this is the preliminary step. That's the in silico analysis. So we have to carry out the in vitro or in vivo analysis for the reliability of the result. That is the future aspect. Also, I would like to expand the protein target that here we only selected the monoamine oxidate. So by understanding the Parkinson's pathway, or that there may be other target proteins. So expanding the targets also and another future aspects I'm considering. I think it is the taraxosterol because it has actually statistically it has taraxosterol and 3-epimethylenic acid because we are considering the comprehensive analysis of both ADMET drug likeness properties along with this docking score. If we consider only the docking score, it will be taraxosterol, but we have to consider as it is for a potential drug target, so we have to consider the ADMET analysis for the safety. So by considering that, it will be taraxosterol and 3-epimethylenic acid.
You have found that the action of multiple compounds will be. Can you recommend the plant as a whole or an isolated product?
If I recommend the plant as a whole, it might have the cytotoxic compound. So we cannot consider the plant or the extraction of the plant whole. So the aim of this study is the identification of that particular target or the ligand protein. So I must recommend the taraxosterol or triethylmethylenic acid alone.
Study will be quite promising in the future.
Yes. Thank you, ma'am. Thank you, sir.
Why you put Amandana plot in the slide? Amandana plot in the slide.
Yeah.
Why?
Because I will select the slide. So this is the Ramachandran plot. So by analyzing this plot, we can understand that most of the amino acids are present in the allowed region. There is only two amino acids, that is the cross mark, which is
2095
compounds. That is present in the disallowed region, which means that this is a stable or feasible compound. So we can use it for the analysis. That is it.
In this area we can in this area.
By to interpret the Ramachandran plot, there is 4 blocks, which is allowed region, and also can be present or that region, and disallowed regions. That will be the right and 2 parts. So here we can see that dots, that is the green color, black color dots, which represent the allowed region, amino acids which present in the allowed region, and there is 2 cross marks. I think it is in the bottom right side. There is only 2 molecules, so that means that only 2 molecules are present in the disallowed region. So this is a feasible compound. So it can be taken for the study. Thank you, sir.
Thank you, Jalwa. Move on to the next presentation. I call Fatima Najjar.
Hello. Hello.
Yes, Fatima Najjar.
Is it audible?
Yes, you are audible. Start.
Can you see my screen?
Yes, you can start.
I'm Fatima Najad. I'm here to present my paper on the topic nutritional profiling of Palmyra palm powder and arrowroot powder versus industrial nutritional supplements. By introduction, we know that food nutrition is the very sustainable for our life and promoting overall well-being. It's involves the consumption and proper utilization of food to meet the body's need. Good health and nutrition are critical for human development. And now we know that many we are consuming many plant and its parts. It gives us many nutrients such as carbohydrates, proteins, vitamins, minerals, and fibers. Here our focus is on Palmyra palm powder and arrowroot powder as nutrient rich natural alternative to the industrial supplements. And our objectives are to compare the biochemical profiles of Palmyra palm powder and arrowroot powder with industrial supplements such as Horlicks and Boost. The parameters we analyzed here is the concentration or the content of carbohydrates, proteins, amino acids, cholesterols, vitamins, and minerals present in these plant materials and in industrial supplements. Here we used materials such as Palmyra palm powder and arrowroot powder. Then we used 2 samples from industrial drinks such as Horlicks and Boost. The collection method of these materials are we collected the powders of arrowroot and palmyra palm from households in Pulpalli, Wayanad, while industrial samples were purchased. And the sample extraction was by 1 gram of each sample was taken in the centrifuge tube. It mixed with 5 ml of 80% ethanol and incubated in taking incubator for 24 hours. Then the mixture was centrifuged and supernatant were collected. We repeated this process 3 times and we obtained a supernatant which we take for the all testers. And here we do biochemical analysis of amino acids, proteins, carbohydrates, vitamins, minerals, and cholesterol. The biochemical estimations we have done here is we estimated the total free amino acids present by using the in-hybrid method, and we estimated the total protein content present in this by using Lowry's method, and then we estimated the total glucose content by using Andron method, and we estimated the protein content by biuret method, cholesterol content by Zach's method, and we also estimated the amount of copper and ascorbic acid present in the samples, all these samples by DNPH method. By this all biochemical estimations, we obtained a result as that carbohydrate present. This table shows the amount of carbohydrate, protein, amino acid, cholesterol, vitamin C, and copper present in these samples. And this graph shows the carbohydrate and protein concentration in 100 gram of natural plant powder versus the industrial nutritional drinks. And this graph shows the concentration of amino acid and cholesterol content in 100 gram of natural plant powder versus industrial nutritional drinks. And this shows the concentration of vitamin C and copper content in 100 gram of natural powder versus the industrial nutritional drink. And the key findings from these studies are carbohydrates are higher in industrial supplements, and the overconsumption of these carbohydrates linked to health risks. And protein, in case of protein, arrowroot powder superior to others. That means in palmer powder, all we are looking, arrowroot powder contain more protein content, and cholesterol it only present in industrial supplements, and cholesterol is not present in palmyra palm powder or arrowroot powder. And vitamins and minerals, plant powders are rich in essential. The industrial supplements have some vitamins and minerals, but it's the plant powders richer in essential nutrients like vitamin C and copper. Then this shows the amount of carbohydrate present in each samples. In palmyra palm powder, 21.3 gram of carbohydrate per 100 gram of sample, and in arrowroot powder 12.26 gram per 100 gram of powder, and for 45 gram of carbohydrate per 100 gram of Horlicks powder, and 33 gram of Boost powder, 33 gram of carbohydrate per 100 gram of Boost powder. Then we are going to the protein. It's in arrowroot powder 4.32 gram per 100 gram, it's highest, and in palmyra palm 1.3 gram per 100 gram. And in we looking for in industrial supplements, it contain in very low concentration. And cholesterol, it's absent in natural powders and present in industrial supplements. Then amino acids are higher in arrowroot powder when comparing to others. And vitamins and minerals, the palmyra palm powder contain 0.017 gram per 100 gram of powder, and arrowroot powder vitamins present in 0.018 gram per 100 gram, and copper in palmyra palm powder contain 0.001 gram per 100 gram, and in arrowroot powder 0.002 gram per 100 gram of powder. The natural from all these studies, we know that the natural powders are nutrient rich and safer for regular users. And the health implications are industrial supplements which are higher in cholesterol and carbohydrate pose risk like obesity, diabetes, and heart diseases. And plant powders such as it's natural, nutrient rich, safer, and it's also cost effective. Then in conclusion, we are saying that plant powders are nutrient-rich and safer alternative to the industrial supplements. Promote their use for addressing malnutrition and reducing health risks associated with industrial products. Natural plant powders such as palmyra palm and arrowroot are nutrient-rich and safer alternative to industrial supplements. They provide essential nutrients like proteins, amino acids, vitamins with no added cholesterols or added sugars, making them ideal for balanced diet. Thank you. This is all about my studies. Thank you Mes Keveeyam College for giving this opportunity to present my paper here.
Hello, Fatima.
Yes.
A nice presentation. Have you published this data?
No, I have not published it.
But if you are publishing, you can hide the brand name as brand one, some code or something like that, because
Okay.
revealing the names as such, the brand name won't be encouraged. That's why.
Okay. Thank you.
You may have to face the legal things then.
Okay. Thank you.
Okay. And about the health implications, you have told that higher amount of cholesterol and carbohydrate in the natural products or the commercial products. The commercial products.
In commercial products.
But your results shows that higher quantity of carbohydrates is in the natural product.
No, carbohydrate higher concentration shown in the industrial products here. Carbohydrate and cholesterol.
Okay, can you show me the slides, please? Okay,
It's
fine.
okay, ma'am.
It's okay, fine. Where was the study conducted?
Parshiraja College, Pulpalli.
Okay, fine. And can you explain the cost effectiveness, because arrowroot and the palmyra powder both are expensive rather than this commercial products, I think, and the availability also. So can you explain?
Actually, in that area, I think like in Pulpalli and in Mayanad, it's not much cost.
Okay. In arrowroot, have you collected the whole rhizome or the powder?
We collected the powder.
From where?
From household.
Household, local market, or just you made it?
Yeah, it's making in home.
Your home only?
Not my home. It's a household product from a native place.
The thing I asked is it is often adulterated. That's why arrowroots have to be.
No. It's natural product. They are culturing. They are cultivating it and they're making the powder from that.
Okay, but this plant need to be authenticated. That's it.
Okay.
Now you can carry over this work and do further analysis so that your study will be benefiting the future generation, because the brands you have recommended or maybe you have conducted the studies already established and used throughout the young generation. So I think you have to do much more and publish this work. Okay, all the best.
Okay, thank you, ma'am.
Move on to the next session. I call Abdul Wahid presenting the topic allelopathy and allelochemicals in pteridophytes, innovative bioherbicides and pesticides from vascular cryptorgans.
Dr. K, Research Scholar, Department of Botany, Aligarh Mystery University. This is my topic. And it was for the allelochemicals, intertophiles, energy biopesticides and pesticides from vascular bacteriums. Basically, this is my research topic for PhD. So I started my PhD just 6 months ago. So I'm just presenting my research ideas about allelopathy intertophiles. As you may heard about allelopathy. Allelopathy, an interference mechanism or a natural political phenomenon in which one plant may interfere with the growth and other properties of another plant through release of some chemicals that may be affect positively or negatively. The chemical responsible for allelopathy is known as allelochemicals or allelopathy. Anyway, it is not a recent branch, but the idea about allelopathy for biopesticide is a recent one. We can trace back to your classes. You mentioned the term, not in terms of allelopathy, in another term, that is in plant interaction for chemicals, had mentioned in his book History of Pandara. And also, the term is coming in 1935 by Hans Mosch. So, allelopathy offers potential for weed control through producing and releasing allelochemical from leaf, flowers, seed, stem, and roots. It's an interference mechanism in which living or dead plant can release chemical substance that inhibit and stimulate the associate plant growth. Then, why there is a necessity of allelopathy? Allelopathy is already an established fact. There is no need to explain the allelopathy and allelochemicals. But in order to understand the, or in order to control the weed, we have to go through allelopathy. In order to regularly interact with the crop plant, we produce chemicals that impact the growth and development of crop plants. That is the important thing. To control the weed population, that weed, you may know that weed that's unwanted plant may interfere the growth of crop plants. So, in order to control that weed, you can go with allelopathy, then to biopesticides. So we are spending billions of years on herbicides to control weeds, to control the weed each year. Overuse of synthetic chemical for weed control worsen the quality of soil, water, and life supporting system, human health, and food. So allelopathy is important in this term. Natural product released from allelopathic plant may help to reduce the use of synthetic pesticides for weed management and therefore less pollution, safer agricultural product as well as lighten the human health concern. For clarification, this is the simple mechanism of allelopathy. You can go allelopathic clone of plant. You may release the allelochemical into soil through bleaching, exhalation, or maybe volatilization, not the soil. Then it will be absorbed by the plants. It may enhance or inhibit the growth. And also, allelopathy may also contribute to the invasiveness, that is the common thing in nowadays, among weed and some predators. Okay, present status, that is, weed are persistently compete with agricultural crop. That's a major threat to the agricultural system that reduce the crop productivity and yield. In India, reduction in crop yield was estimated as 31.5% by weed, whereas economic loss due to weed was estimated as INR 28 billions about 2 decades ago. In another study conducted by maybe another report from National Research Center for Weed Science, it was reported that loss in agricultural productivity due to weed amount to 1,015 billion per annum. So these results indicated that among the major constraints in agricultural sector, weed alone contributing a major threat to the sustainable agriculture. So synthetic herbicides, we are often using synthetic herbicides to reduce the impact of weed population, which comprise more than 45% of all pesticides sold worldwide and have been primary tool used to control these weeds. Even though these substances have demonstrated instant effectiveness in suppressing various weeds, repeated and prolonged use may have a detrimental ecological effect. So, allelopathy seems to be an alternative tool to chemical-based agriculture to reduce the impact of these toxic herbicides and pesticides on the environment. And the purpose of system is to develop toxic-free food for humans and animals in a sustainable manner. So, in line with allelopathy, another potential approach is not another potential approach. It's through allelopathy we can develop bioherbicides, and already there is more than a dozen of bioherbicides from bacteria, fungi, plant extract were discovered, but 95% are not available or are a limited availability. So there is a lot of bioherbicides available through various means, maybe through bacteria, fungi, or plant extract, but what is their present status is either unavailable or have a limited availability. So in the present circumstances, and one thing, allelopathy is a lot of studies conducting on allelopathy, basically in flowering plants, such as angiosperm, maybe you can say that gymnosperm, but we can go for another source. That is what is next. We can go for peridophyte vascular peridotams. So the allelochemicals, such as that is basically the secondary metabolite, not all the secondary metabolites, but some category of metabolites such as polyphenol, phenol, terpenoid, and glycosides, and so on. So phytochemical analysis of foam revealed many secondary metabolites such as flavonoid, steroid, alkaloid, phenol, triterpenoid compound, and different amino acids and fatty acids, etc. And also, more than 157 species of peridophyte, especially fern, have been recognized as an exotic breed, which around 39% have the property of invasiveness, even though peridophytes are ecologically and economically important plant group. So this is the basic idea why I am taking peridophytes instead of other group of plants such as major angiosperm and gymnosperm. It has a potentially a large amount of secondary metabolites and it can be used. Other maybe it is still using in other bioprotencies such as antibacterial, antiviral, antifungal, but it is not commonly using it for maybe for allelopathic studies. So these are the materials and methods to establish the allelopathic effect and its role in invasiveness. Also the bioherbicidal and pesticidal potential. We have to collect the plant material and properly wash it and dry it. That is means shade drying. And after that, you have to make it powder. Then you have to make different solutions of different concentration. Then we go for a petri plate experiment that is using seed germinator to to provide the optimum conditions. Then you have to go to greenhouse conditions that should be carefully taken to avoid the competition and seedless. So along with allelopathy, there is another mechanism of interference that is competition. So you have to avoid all the elements that is related with competition from allelopathy. Then you have to go to field trials in agriculture. Then because selected allelopathic source plants show allelopathic effects against weed of major crop rather than crop itself. There are different kinds of allelopathy maybe in terms of plants, maybe crop to crop or crop to weed or weed to crop or to weed to weed. So we have to use the crop to weed allelopathy to analyze whether it shows any bioherbicidal potential. We can go for first for primary phytochemical analysis using some chemical test, or you can go to GCMS, HPLC, FTIR for phytochemical analysis and quantifications. Then individual extractions and separation of each secondary metabolites. If necessary, you can go for NMR spectroscopy to analyze the structural details. Then go for petri plates and greenhouse experiment. Then field trials in different climatic conditions. Then future prospects and conclusions. Natural compound derived from allelopathic plant may lessen the need for synthetic pesticides to control weed, which could cut pollution, make agricultural goods safer, and ease worries about human health. Therefore, investigating the possibility of using plants with high allelopathic activity to control agricultural weed is worthwhile. This study can be further elaborated to identify crop and weed vulnerable or resistant to allelophysical and to find a viable bioherbicide for sustainable agriculture. In the same way, we can find the biopesticidal properties. Thank you.
Actually, you have a very good aesthetic sense because your slides are very well organized and presentation, and you have done a very thorough and extensive groundwork regarding your future.
I have just started my research in 6 months ago.
Study will be very much, you are very much involved in your study, so hopefully it might be very good status for your career. And which period of fight are you planning to select?
Now I'm conducting experiment in primary portfolio. I got some result in specific place, but I didn't go for a. I will, but I didn't go for a greenhouse other experiments.
Okay, then which one seeds? Which seeds are you selecting?
Seeds. Seeds.
I'm
Yes.
thinking the vital constituents in these pteridophytes. I think will it affect the germination or the growth patterns?
Both parameters. We are analyzing growth parameter, biochemical and physiological analysis. Then I have used for primary portfolio. I have used Vigna and Pisum, then wheat and brassica.
And you have selected binary.
Primary, yeah. Means it's
Actually, primary is arboreal, right?
Yes, arboreal. It's only available here in Kerala. So there is a difficulty to collect and carrying that. Anyway, it's showing good result. May I have to choose plant instead of.
You have to isolate the vital constituents from this particular plant and then apply on to
Okay.
So, I can just a recommendation. You can select Marsilia or Solar because
Why already
these are
collected
organically
Marsilia?
screened in
Yes, ma'am. I already collected Marsilia. You returning some within a week back to university. So I'm collecting. There's a difficulty because it's only available in monsoon season, most probably in watery conditions. Still, I couldn't find a nowadays. So I have to wait for some.
Can you predict these plants identified as seasonal? So they might be secreting these chemicals only seasonally. Will it be persisting throughout the season?
So will be a bias if I have to say. I don't know.
So you can check that because these chemicals are released into the natural environment, and these plants are seasonal, but the crops might not be seasonal.
Yes. It's a very different climate in North.
And other parameters will be can be kept as a constant. Under the control condition, you can check the persistence of this vegetable. The next is what kind of interaction is there? Is it a positive one or a negative one according to your findings?
Both positive and negative because I'm previously I worked in the college for a while, up to 2.5 years in Marcus assistance. So at that time, for a religious student, I conducted experiment using Euphobia atrophica in Santa means Vignan. It shows a positive result, and in also in supervitation. So it's showing some positive result. It's germinating if you increasing the concentration of extract. So comparatively, enhancing result means increasing the growth. Still, it's not common, but increasing results are more pronounced.
The phytoconstituent secreted from one particular plant might be positive for some, negative for some. That's what you have found. Nice. And you have mentioned about the biopesticide problem. How can you create
Yes. And still, for conducting biopesticide, I need a man from zoology. So I'm still searching for that to conduct along some experiments because especially in insecticide. So sorry, I just against insects. So that's hopefully.
So you're actually in urge of collecting some, finding out the natural remedy for herbicide as well as pesticide. Good work.
Thank you.
All this.
Yes, nice presentation. Whether you know Dr. Rajesh from Connecticut. He's specialized in dermatology. Now it is Guruvayur College, Connecticut. So means I think it's up to you. You can interact with him because that will be better to and whether you are going to check the effect on invasive plants.
invasive plants. There are some studies going on not my seniors are conducting some studies on invasive species. They are, but I'm not much interested to go with the invasive species.
No, if your plant extract is enhancing the growth of invasive species, then it might not be
That's fine.
desirable.
Yeah.
I once have contacted Santosh Nandi.
No, yeah.
I have contacted.
On to the next session. I call KR Anisa.
Am I audible? Hello, am I audible?
Yes, yes. You can start now.
Is the slides visible?
Yes, yes.
Okay. Good evening, everyone. I'm Anisa, research scholar in the Department of Aquaculture and Fishery Microbiology, MES Ponnani College. Today, I'll be presenting my research on the topic Evaluation of Virulence Factors among Multi-Drug Resistant Bacterial Isolates from Central Coast of Kerala, South India. This study sheds light on the growing challenge of antibiotic resistance in the region and its potential impact on public health. To begin with, the inadequate selection, misuse, and overuse of antibiotics, particularly in agriculture, aquaculture, industries, etc., can lead to resistance in various bacteria, and this will make the treatment of infections more difficult. The virulence factors such as hemolytic activity, cell surface hydrophobicity makes the bacteria able to cause infection. Here I have selected the coastal water because they are subjected to a high rate of environmental pollution and they can also serve as a natural reservoir of antibiotic resistance and also a conduit for the spread of this resistance. The bacteria also evolve mechanisms such as biofilm formation, which will again make the treatment of bacterial infection more difficult. And also the production of ESBL enzymes that has the ability to hydrolyze the beta-lactam ring of the penicillin group of antibiotics. And also it work against the third generation cephalosporins like cefotaxime and ceftazidime. This is also a major factor in antibiotic resistance. So, I have formulated my objectives as these two to study the prevalence of antibiotic resistance and multi drug resistant nature among the bacterial isolates from coastal water and sediment origin, and then to determine presence of ESBL production, biofilm, and virulence factors, hemolytic activity, and cell surface hydrophobicity among the multi drug resistant bacteria isolated. So my sampling sites were Ponnani and Chittuva harbors and Talikulam and Arikode beaches that cover both Malappuram and Thrissur district. I have collected water and sediment samples and I have done total plate count and then isolated colonies were gram stained. Then I have done the antimicrobial susceptibility test which is named disk diffusion. Here is the procedure for that, and I have performed it on the Muller Hinton agar plates, and the bacterial isolates were classified as susceptible, resistant, and intermediate to each antibiotic based on the result. I have used 16 commercially available antibiotics for the study. Then I have calculated the multiple antibiotic resistance, that is percentage MAR and MAR index. Then isolates that are resistant to 3 or more antibiotics of different categories can be grouped as multiple antibiotic resistance. If a bacteria is having MAR index greater than or equal to 0.2, it indicates they originate from a high risk source of contamination where several antibiotics are used. Then I had gone for the calculation of percentage incidence of antibiotic resistant bacterial isolates from each sample source. Now coming to the ESBL production. For that ESBL production, I have followed the double disc diffusion test, which have two tests, a presumptive test and a confirmatory test. As you can see in the picture, an increase of 5 millimeter in the zone diameter of the combination disc with clavulanic acid. It will confirms the ESBL production. ESBL is the extended spectrum beta lactamase enzyme. Then I have checked for the biofilm formation in the bacterial isolates. This is a procedure for the biofilm formation checking by tube method. You can see in the picture a strong biofilm forming bacteria and a moderate and a non-biofilm. You can see the differences in them. Then I have gone for the detection of the virulence factors. I have checked here hemolytic activity and cell surface hydrophobicity. For hemolytic activity, the plate hemolysis tests were done, and you can see the presence of a zone of complete lysis of erythrocytes around the colony in case of a positive plate. And in case of cell surface hydrophobicity, I have done the salt aggregation test. Here, the bacterial clumping can be seen if the bacterial isolate is hydrophobic. Then the selected isolates that have short positive result to the ESBL production, hemolytic activity, and cell surface hydrophobicity were undergone molecular identification by the 16S rRNA gene sequencing at Genspect Private Limited, Cochin, and they were identified up to the species level. And coming to the results, these are the results of the bacterial analysis. Here, a total of 78 bacterial isolates were obtained. Here we can see the table of gram staining and types of bacterial isolates. Coming to the percentage MAR table of gram negative and gram positive isolates from Ponnani and Chetua harbors. In left side, you can see that of Ponnani harbor, and in right side, you can see that of Chetua harbor. Here, 78.26 percentage of isolates showing percentage MAR greater than or equal to from Ponnani harbor. And in case of Chetua harbor, it is 93.75. You can see in case of Ponnani harbor, the bacterial isolate number 10 is having the percentage MAR equal to 50, which means they are resistant to half of the antibiotics that I have used for my study. I have used 16. They are resistant to 8 of the antibiotics. Coming to the percentage incidence chart, in case of Ponnani harbor, nitrofurantoin is showing the highest percentage of incidence here. 91.3, and in case of Chetua harbor, amoxicillin is showing highest percentage of incidence of antibiotic resistance. Here amikacin, streptomycin and tetracycline is showing 0%, which means all the bacterial isolates from Ponnani harbor were susceptible to these 3 antibiotics. In case of Chetua harbor, it is ciprofloxacin. Here it is the percentage MAR table of the gram negative and gram positive isolates from Thalikulam and Areekode beaches. Here again, in case of Thalikulam beach, 94.44% of isolates showing percentage MAR greater than or equal to 20. And in case of Areekode beach, it is 90.47. Here is the percentage incidence graph again. In case of Thalikulam beach, highest percentage incidence is shown by amoxicillin, ceftazidime. And in case of Areekode beach, it is shown by amoxicillin again. Here, susceptibility was shown by amikacin, trimethoprim, streptomycin, and gentamicin. Here, it is ciprofloxacin again. Now, coming to the results of ESBL production and biofilm formation, only the 31 isolates out of 78, 31 isolates were confirmed as ESBL producers after the double disc diffusion test. And surprisingly, all the ESBL producers showed multiple antibiotic resistance, and ampicillin was common among them. Among the 78 total isolates, 42 were strong biofilm forming ones, and out of it, 31 were confirmed ESBL producers. And in case of virulence factor, 24 isolates showing MAR index greater than 0.4 were screened for the virulence factors. Among them, 10 were hemolytic and 12 were hydrophobic. 8 isolates showed both hemolytic and hydrophobic activity. So from this 8 isolates, 2 of the isolates from Ponnani harbor, which is having MAR index 0.4, were undergone the molecular identification by 16S rRNA gene sequencing, and they were identified as pathogenic species like Vibrio, Alginolyticus, and Bacillus cereus. So coming to the conclusion of my study, the presence of pathogenic bacteria in the harbor and surrounding areas poses a significant public health risk, especially since the local population uses the coastal water for various purposes. And the prevalence of multiple antibiotic-resistant strains, including those producing ESBLs, further underscores the need for ongoing monitoring. The present study reveals that apart from clinical isolates, environmental isolates of bacteria also have become resistant to various antibiotics, and it brings forth the need for stringent regulation in the procurement and application of antibiotics. These are my references. Thank you. Hello.
Anissa, very relevant work. You have presented really well.
Thank you, ma'am.
Anissa, can you hear?
Yes, I can hear.
Okay. Have you gone for any identification of the bacterial strain?
Yeah, I have gone for the molecular identification of 2 of the isolates.
Okay.
And
Is
they
there
were
any new
identified
species?
as Vibrio alginolyticus. No new species not were recorded, but it was identified as Vibrio and Bacillus cereus. That means they are pathogenic.
How many pathogenic bacterial strains have you identified?
Only 2.
2 pathogenic. Okay. Is there any reports of pathogenic infection in that area?
No, not from environmental samples.
Not so. No need of using antibiotics against these strains, like.
Yeah, I think a new class of antibiotics should be discovered.
Okay. What are your recommendations for the medical practitioners who are prescribing
Uh,
amoxicillin? Because in 3 of the sites you have done your study, amoxicillin is
yeah,
finding to be resistant. Right? All the strains are resistant against amoxicillin. So
yes, yes,
is there
yes, ma'am.
anything? Okay. Is there
If we
anything?
can develop any alternative therapeutic methods like, yeah, they can develop any alternative therapeutic methods.
Okay, fine. So, what is the future prospect of your study? Because there are no pathogenic fungi,
Um,
I mean, pathogenic bacterial strains.
Bacteria.
There are no
Yes,
pathogenic
ma'am.
bacterial strains. Only 2 bacterial strains, like, and no reports of pathogenic infection.
Yeah, I
So,
have done the molecular. I have done the molecular
what is the point in using
identification of only 2. Rest I have to identify.
only 2 you have
No, I have
identified, right?
78 bacterial samples with me. Yes, I have
Ah,
identified only 2 of the bacteria, and the rest I have to undergo molecular identification.
okay, then, okay, fine. So, you can go for antibiotic resistance activity in all the pathogenic strains.
Yes, yes, ma'am.
Then you can recommend the medical practitioners to avoid yes, prescribing amoxicillin also.
Yes, hope so.
Okay, nice book, Anissa. Nice book.
Thank you. Thank you.
All the best.
Thank you, ma'am. Thank you so much.
Let's move on to the next presentation. I call Abhishek Nayak to present his topic.
Abhishek Nayak. ഇത് കംപ്ലീറ്റ് അല്ലല്ലോ വർക്ക്. ഞാൻ ചെയ്യുന്ന ഭാഗത്തിന്റെ ഒരു ഭാഗം എന്ന് പറയുമ്പോഴും ചെയ്യുന്ന വർക്കിന്റെ ഒരു ഭാഗം എന്ന് പറയുമ്പോൾ കംപ്ലീറ്റ് സിനാരിയോ പറയണ്ടേ.
Good evening. My name is Abhishek Nayarar. I'm a 4th year student in Integrated Botany with Computational Biology, Mes Keveeyam College. My presentation is on a pilot study on allelopathic effect of Vedalia trilobata on Vigna unguiculata cultivar Kanakam. Allelopathy is a biological phenomenon by which a plant produces one or more biochemicals that influence the germination, growth, survival, and reproduction of the surrounding plants. Allelochemicals are often negative effects that helps in plant defense against herbivory. Allelopathy is also a great alternative for weed management in various prospects. In this project, it mainly aims to understand the allelopathic effects of Vedalia trilobata, focusing on Vigna unguiculata cultivar Kanagavani. The objectives of this study was to compare the effect of allelochemicals produced by Vedalia trilobata on the germination of seed, leaf width, leaf length, root and shoot length of Vigna unguiculata, CV Kanagamani, and also to study and compare the effect of allelochemicals of Vedalia trilobata on the chlorophyll content of Vigna unguiculata, and also to create awareness among the local farmers about the deleterious effects of this weed. Materials and methods. The weed for making the extracts were collected from Tanur, Malpuram, and after which they were dried for 30 to 35 days. It was sun dried, after which it was completely powdered for preparation of the extracts. The seeds of Vigna unguiculata cultivar Kanagamani was collected from the pulses crop division of RARS Pattambi. The aqueous extracts were prepared for 1%, 2%, and 3% concentrations consecutively by using 2 gram, 4 gram, and 6 grams of the powdered weed on 20 milliliters of distilled water, and then which was separated using filtering for extract purposes. After which we had conducted the Petri dish bioassay. The Petri dishes were lined with cotton and 10 healthy seeds of each were placed, making triplicates with a control of distilled water, and then 3 of the concentrations of 1%, 2%, and 3% with a 30 ml of weed extract. And after which the observations were made for 7 days. The measurements taken were of the root and shoot length, leaf length and leaf width, germination percentage, seedling vigor, and also the chlorophyll content. The chlorophyll content was determined using Aaron's protocol of 1949, in which 0.05 gram of leaves were used for making by 10 ml of 80% chilled acetone. After making the extracts, these were used in spectrophotometer at an absorption of 645 nanometers and 663 nanometers for calculating the content of chlorophyll A and chlorophyll B exactly. The results of this experiment showed an adverse effect of the weed extracts of 1%, 2%, and 3% solutions in comparison to the control on the germination rate, leaf length, width, shoot growth, and root growth, and also the chlorophyll content. This was the data given for the germination rate of various concentrations of weed extracts. You can see that in control, we have a germination rate of 80%, whereas there's a significant reduction from 1%, 2% to 3% concentrations, where finally giving 3% concentration of 56.67% of germination rate. The concentration of weed extract on leaf length was also an adverse effect, with the control giving much more length when compared to the concentrations of 1%, 2%, and 3%. From the day 3 to day 7, these were noted. And for the leaf width also, the control had given a 1.42 centimeters of leaf width on the final day, whereas the 3% concentration with a significant reduction gave only 1.07. The shoot growth was also adversely affected from the control to concentration 1%, 2% to 3%. On the final day of day 7, we got a shoot growth of 23.8 centimeters in the control, whereas the significant reduction caused the resulting of shoot length of 13.3 centimeters on concentration 3% on the final day. In the root growth, the length of the root on the final day that was taken, that's the 7th day, had a significant reduction. From 8.3 centimeters, it had gone down to a 3.5 centimeters of root length, 3% concentration on the final day. And for the chlorophyll content, the control gave a number of 20.577 and concentration 3 was 15.5345 values. Here we could not see a significant reduction in the amount, but it was observed a mild reduction in the chlorophyll content. This is the image showing of different days of work. Here we can see there's a reduction in the growth from day 1 to day 7, where we get a control of fully grown plants in concentration 3%. There's a much reduction is observed. Here too in the root growth, where the control gave more root growth, and there's a significant reduction observed from concentration 1%, 2%, and to 3%, giving a less root growth. In conclusion, we can say that the result of the study clearly indicated negative allelopathic effect of the weeds. The germination was least in 3% when compared to the 1% and 2% concentrations, and also same with the root and shoot growth, which was found significantly reduced in presence of weed. The controlled condition germination and root shoot length is higher in Wedelia trilobata extract conditions, and the chlorophyll content in 1%, 2%, and 3% had a reduction while compared to the control used. So this study can conclude that the growth has affected in 3% concentration than in the 2%, 1% concentrations. This shows the drastic effect of the weed on germination and growth of the Vigna unguiculata cultivar Kanagamani. Thank you. Thank you.
So why did you consider the dried product instead of fresh?
Huh?
You have used the dry product, dried leaves.
Huh.
Which part you have used for the study?
Whole plant was used for the study.
Dry form.
Huh.
Why did you go for why didn't you go for the fresh instead? Fresh part.
No, at first we planned to use this powdered weed only, so we went with that plan.
Because then because of weeds and not growing along with the you have done for the fresh plant part yours.
Ma'am, there has also been uses of Wedelia trilobata as herbicides and herbicides. So in that forms, it was mostly used as powdered forms along with this water. So we had to test whether it can be used as a better herbicide or not. So we saw a reduction also in the crop plant. Yeah, Wedelia is an invasive species. No, phytochemical analysis was not done on this.
So your prediction is what might be the principal component which is responsible for the allelopathic of this?
And there has been various studies on this Wedelia trilobatus phytochemical. So many flavonoids and terpenoids were present in this. So those may be the reasons for it.
And this plant be employed in beneficial use and might playgrounds.
Now, if we were to work more on this target specificity of this plant, then we can go on this field. But till now, there's no target specific results for this. Any crop grown near to this field is shown a reductive rate. It was taken for 7 days. 7 days, and the measurements were made from day 3 onwards. Yeah.
Check the heat also, then it will be better. I suppose you can carry this over for future studies.
Vijayashree K for presenting her topic.
Good evening, everyone. My name is, I hope I'm audible. My name is Vijayashree K. I'm a research scholar from the Department of Aquaculture and Fishery Microbiology, MES Panani College. And the topic for the presentation is macroinvertebrates and fouling organisms on litters and cage fish farm in the Panani estuary. Introduction. The stationary or sedimentary character of benthic invertebrates makes them good markers of aquatic ecosystem health and environmental changes. Marine fouling macroinvertebrates develop colonies or adhere individually to submerged surfaces. These organisms cause biofouling, which damages vessels and infrastructures. Increasing anthropogenic litters in the maritime environment offers a substrate for marine species, and this litter facilitate the transport of fouling organisms. Human activities, including urbanizations, industry, and ports, have released many contaminants into marine habitats. And oysters, clams, mussels, and barnacles are categories of fouling organisms. However, not all macroinvertebrates fall into the category of fouling organisms, which I particularly say that I have collected macroinvertebrates which are not fouling organisms. Objectives for the study is to identify the type of macroinvertebrates and fouling organisms present on litter and cage culture system in the Panani estuary, determine the month-wise variation of organisms from the selected site, and for the assessment of marine fouling macroinvertebrates inhabiting on litter and cage culture system in Panani estuary. Materials and method for this study, I have selected 3 distinct sites for its uniqueness. For site 1, which is selected in Panani Harbour, which there is immense of fishing practices are happening, and site 2nd is a cage culture farm with 20 units culturing latest calcarifer and Retroplus suratensis. There is a culture area. And site 3rd, which is located in a tourism focused area on Karma Road, which is mainly focusing on tourism and other activities. Samples were collected on a monthly basis from October 2023 to March 2024. And the collection of species and preservation, sample species are collected by handpicking and by stripping with a sharp knife, and the collected species were sorted and identified and preserved for the further analysis. And the result, the specimens or colonies collected from the site from two phyla, which is mainly mollusk and arthropoda. And so 20 species were documented on marine litter and cage installations. The quantity of distinct colonies recorded per litter ranged from 1 to 200 present in all litters and everything. And mollusca is the dominant phyla in 3 sites, followed by arthropoda in site 1 and site 3rd. Amphibalanus amphibrytes, that's the barnacles, was most abundant at site 3rd, while site 2nd predominantly hosted Medulastigata. It is itself a university species, and Sarcostria spatulata in site 1 species like Castosphere virginisa and Hyotis sinensis. Medulastigata, because as I said, it has an invasive nature, are dominant and seen only associated with the cage fish farms. Amphibalanus amphibrites are dominant in phyla Arthropoda in site 1 and site 2nd, followed by Paraseserma licatum. And all the 3 sites show dominant fouling organisms like Amphibalanus amphistrite, Medulastigata, Sarcostria spatulata, and Castosphere virginisa. And these are the table for the representation of all the species. And accordingly, month-wise variations have reported, and the status also for the sea, common, abundant, and rare. And it is from the harbour side. And the site 2nd is cage fish farms, which is mainly seen fouling organism, that is Sagostrase, Patulata, Crassosauria, and Mittellastigata. And site third is Karmarol, which is the tourism area. And the third, it is a percentage representation. So gastropod is the main species, class, which is present in side 1 and side third also. And fouling organism, these are the fouling organisms which is present in these 3 distant sites. The discussion and conclusion and the preliminary research indicate a rich diversity of macroinvertebrates, especially mollusks and arthropods at all sites. And fouling organisms were abundant on marine litter, including plastic bottles, wooden debris, fishing nets, and ties, as you can see there in the first slides and all, as well as on structures within the cage culture system. Marine debris carries fouling organisms, including invasive species that potentially have ecological impact on the marine environment and many threaten biodiversity. Mittellastigata threaten livelihoods of local fishes by disrupting traditional fishing and aquaculture practices, because this cage culture practice is happening there in Ponani estuary. So the Mittellastigata is creating problem for the fish farmers, which is more focusing on cage culture farms. And some species exhibit seasonal presence due to the salinity, as I've presented that table there. Sometimes December only, some months only they're predominantly seeing, maybe because of the presence of salinity variations. And these are the reference for the study. And thank you. Thank you, sir.
Please go to methodology.
Because this is not my actually this is part of my PhD work. We have done that is for a PG student, so I forgot it because we are working with Mittellastigata. So that the thought comes like fouling organisms will be there, so we have to find out that, and we have paper also. From that we have to produce a paper. That's why.
Stripping the sharp knife.
Yeah.
Okay, but in your title you collected it from litter sampling.
Yeah, litters also. Yeah, but collecting like handpicking only, like litters, like the plastic bottles and everything, we have to collect with the hand only. So that otherwise, from the sediment we have to use the grab and all. So I'm not focusing on the grab, like the sediment.
Unit analysis and litter samples.
Litter samples and whatever the fouling organisms are present.
What about the tide? Use that
Tide, low tide, sometimes, yeah, low tide. Low tide, yes. Then only that possible.
Okay, then this is arthropod sample. Only one species.
So no, I know predominantly which is saying I have got it that only. Yeah. No, sir. Actually, the thing is like Ponani Harbor, and they have different characteristic of like when you go for harbor, you'll collect that. You can easily collect it from without the water area, and when it go to the estuary, like the cage culture farm and all, we have to like collect it from the net or not. So that is different. Actually, it is in a one stretch, but we have to go through like different.
Three sides are there.
Yeah.
Each side, how many sample forms are there?
Like.
Three sides are there.
Yeah.
Locations are there.
Yeah.
Each side you put how many samples?
Yeah, 20 species are collected, but the number will be different. Like sometimes they'll be in colonies and all. So that's why I mentioned in a one species like that. So the number if I go to is 3,000 more than that. It is not countable. So I have that. That's why I created. Yeah, thank you. Yes, ma'am. Thank you.
Can you explain why the estuarine? Is there any difference in the estuarine and marine?
Actually, the marine will be like completely saline will be like 31 above that. Sometimes, but when it comes to estuarine and in monsoon seasons, we can see the variations. Like
what was the duration of estuarine?
6 6 months only because we have yes, but month wise only, like October to December 2024. That's I didn't go for that seasons and all. Yeah, but then we have to say it it's like that. But I just you know I've just the months are no like for a PG. Yes. So we are studying on that only, like further molecular studies now. But now I'm just disseminating that. Yeah. Yes, definitely. Yeah. Um, post monsoon only, like so monsoon season, the variations, salinity variation will be changed completely, so they can't survive that, especially with Mitella stegata and all. Mitella stegata, actually, I get this idea when I got, because I am associated with cage farms. So these Mitella stegata is an invasive species, and it is first observed in 2019 in India, which is in Vembanad Lake, Kerala. So from there, these Midlasticate is creating problem to cage culture farms, like they are adhering to net cages and all. So regular removal of this is strenuous job. So that's why from there we have. Yes, but the thing is, some people it is edible, but we are not consuming that. Yeah, Ponani people are not consuming that, but when it goes to Calicut and all, they are consuming. That's the thing. No, that's why we use knife. Yeah, yeah, sharp, any sharp edges we can remove that. Otherwise, it'll create suddenly. Yeah. Thank you. Thank you.
Thank you, Vidyashree. Next, I call Fasila, presenting her topic.
Good evening, everyone. My name is Fasila, Senior Research Fellow, Central Marine Fisheries Research Institute. My study topic is study on feeding ecology of two gastropod species, Clypeomorus batylariformis and Planaxis sulcatus from a rocky intertidal area. The intertidal zone are one of the most important marine ecosystem, and gastropods are the most representative components among the macrofauna. Genus Clypeomorus and Planaxis are gastropods under the superfamily of Cerithidia. Both species are common in intertidally and characterized with cluster distribution pattern on larger rock crevices. Objectives of the study: to determine the gut composition of Clypeomorus batylariformis and Planaxis sulcatus and to characterize radular structure. Importance of the study: despite the study on distribution and abundance of the family Cerithidae and Planaxida were done from the Indian intertidal region, little is known about gut composition, over it is considered as macrophagous, herbivores, and detritus feeders. This study helped in determining species-specific grazing preference and important differences in food selection of these studied species, even it is considered as same functional feeding guild. These are the study site. Then materials method includes to determine the relative numerical abundance of food items in gut content in 1 ml. A surgery crafted condensate was used for determining the gut components, and the abundance of countable gut content was evaluated based on the formula N equal to P into C into 100. N means number of each food items in the whole gut. P means total number of food items counted in 10 fields. And C is the volume of final concentrate of the gut samples in 1 ml. Then gut content was enumerated using counting chamber by using Carl's axiola equivalent microscope. For quantitative analysis of gut contents, the percentage of numerical importance of each countable gut content were determined. Then characterized radular structure by scanning electron microscopy. Firstly, radula treated with 90% potassium hydroxide for removing the adherent tissue and dehydrated by a series of increasing concentration of alcohol. Then radula was mounted on a double stick carbon paper which was stuck on a glass slide. Then some analyses were done at the sophisticated test and instrumentation center result. These are the study species. First one is Clypeomorus batylariformis and second one is Planaxis sulcatus. Results and discussion. Gut contents include macroalgal fragments, diatoms, blue-green algae, and algalspore and detritus. Macroalgal fragments composed in Clypeomorus batylariformis with 18.71%, while in Planaxis sulcatus 4.28%. In diatom composition, in Planaxis sulcatus was 10.96%, and in Clypeomorus batylariformis was 9.96%. Blue-green algae composition were 8.6% in Clypeomorus batylariformis, while in Planaxis sulcatus was 10.97%. Detritus were higher in both species compared to other gut contents. Previous studies on the family Cerithidae and Planaxida depicted that mostly are detrivores, which grazes on microalgae covering rocky substrates. These are a table showing the gut contents of two species. These include macroalgal fragments. In this include foliostripe, filamentous stripe, and others. Then diatoms components include several genus of diatoms: Coconis, Navicula, Cyclotella, Acnanthus, Dermatophora, Melosira, Pinnullaria, and Phosphonodiscus. These percentage of numerical abundance is proportionally different in both species. In the present study, macroalgal fragments are also included rather than microalgae. Filamentous type macroalgal fragments were common in both species, but higher in Clypeomorus batylariformis, and foliostripe fragments present only in the diets of Planaxis sulcatus. In diatoms, Coconis species and Navicula species dominate in both species as numerically. There are reported diatoms have been shown to be important food source for a number of gastropods, but there is no diatom preference for feeding of these gastropod species were detected. Underwood in 1979 suggested that it is difficult to study the preference of food by macrophagous generalist feeders, which feed on any macroalgal and microalgal material that can be accessible for it, and spolix must contain in microalgal film they eat. Some analysis revealed that radular ribbon of both species were relatively long and characterized with long teneoglossant radula, and there are differences in structure and shape of central rachity and lateral tooth of both species. The rachid tooth is scorish in shape in Planaxis sulcatus and pentagonal in Clypeomorus batylariformis. In Planaxis sulcatus, the lateral tooth has broad central cusp with about 2 small denticle on the inner side. In Clypeomorus patellariformis, lateral tooth is rhomboid with long twisted basolateral projection inserting onto basolateral membrane. In Planaxis sulcatus, the marginal teeth are long, narrow, and expanded to claw shape at the tip. In Clypeomorus patellariformis, marginal teeth are long and spatulate, rounded at the bases and having curved spoon-shaped tips. This is the scanning electron microscopical image of the radial structure of Clypeomorus patellariformis and Planaxis sulcatus. Then variation in the consumption of food by herbivores, gastropods for food probably depend on radiolar shape, structural differences, and feeding behavioral difference within the individual of the species. These images showing the gut content of these two species. This is the in situ observation of the grazing by Clypeomorus patellariformis. I could not see that in the gut content in the Clypeomorus patellariformis. This like grazing may lead to breakage of the algal thallus in the field. Conclusions: Clypeomorus patellariformis and Planaxis sulcatus are algivorous microphagous feeder. Structural differences in radula of each species influenced on macroalgal food preference than microalgae. So the grazing of herbivorous gastropod species on macroalgal community is species specific, even the species included in the same functional feeding guild. Thank you.
Ecological significance of this book.
Yes. Ecological significance. It can reduce the macroalgal abundance by grazing in the intertidal zone.
Whether that will affect the population of other diversity?
Not much negative effect on the algal community, but someone balancing it can reduce the macroalgal abundance in that ecosystem.
The sampling and experiment work from
Yes. Calicut. Thank you. Thank you. 20 samples per month. This is the 2-year study. This is a small part of my study. Size one side. Yes. Ticcodi rocky indentellary, Calicut.
That's why you're getting that particular.
Yes.
Can you mention that you've shown picture of macroalgae which you have calculated for the but
Okay.
can you mention the name
From the gut content species.
not that macroalgae?
Yes.
Shown a picture.
First one picture.
No, I've shown a picture of macroalgae.
I microalgae. Okay. This one. First one is Spinularia. This is not. This is Colarpus atralidis. Colarpus species. Sorry, I don't know recently, but there were a study related to coral reef bleaching. Haladharan, sir. Coral species. There are so many coral species. Okay. This is used as ecological modeling studies.
Can you do an ecological?
This is how much abundance of these two species may affect the which category of macroalgae in a particular ecosystem.
That was what, sir?
Right.
Ecological significance.
Okay, then.
So, whether you have went for any workshop regarding modeling, visual modeling?
I did not attend yet modeling studies, but I hope I can do it. Yes.
Is there any environmental conditions which is affecting the
Seasonally different grazing effect. This is pre-monsoon is higher than the post-monsoon season. The intensity, yes, yes.
That formula from where you got the N is equal to B into C.
Yeah, yeah. Abdus Saman, I did not include in this.
You will include that in the slide.
Abdus Saman 2020.
20.
Yes.
20.
Yes, yes.
Previous slide.
Abdus Saman in 2020. Thank you.
Now I invite Musna please to take a vote of thanks.
Good evening to everyone. It's my privilege to propose vote of thanks for the remarkable international conference on integrating disciplines for sustainable development. First and foremost, I express my heartfelt gratitude to our chairs, Dr. Archana ma'am and Dr. Anis sir, for the inspiring leadership and valuable insights that enrich this platform. And I also thank to all participants, panelists and speakers who contributed their expertise and ideas, making this platform more meaningful. And a special thanks to our organizing team, my colleagues, for their hard work behind the scenes and a smooth ensuring of this program. Lastly, I express my gratitude to each and everyone. Thank you.